Predatory mites. Predatory mites can provide natural pest control by eating unwanted pests. These beneficial mites are one way to maintain pest population at non-damaging levels, known as biological control. Moreover, predatory mites occur naturally in varied habitats from agricultural fields to small backyard gardens. It is the best to encourage native population through recognized conservation practices. These practices include scouting for pests and predators and using soft pesticides rather than broad-spectrum chemicals. Reproduction of predatory mites. The acarina are all dioecious. In most groups, the males can be distinguished from the females, even though males and females are similar to each other. Fertilization is internal, but the methods by which the spermatophores are introduced into the female reproductive system vary considerably. Some mites develop young parthenogenetically, but none has been shown to be exclusively parthenogenetic. Moreover, mating behavior and phytosidae has been studied in several species. Geotonimpal females of Metaxilus occidentalis, for example, produce a contact sex pheromone that causes male to guard them and mate them with shortly after they molt to adulthood. The image shows the schematic representation of mite life cycle reproduction. Males and females are dimorphic, with males being about half the size of adult females. Indirect sperm transfer involves deposition of sperm packet on the substrate by males. In some cases, the spermatophores are deposited in groups. Females then pick up spermatophores and place them in their sperm receptacle. Female phytoseeds that are unmated do not produce eggs. Yet, eggs appears based on cytological evaluation to have two different chromosome numbers, which made people assume that they are haplodiploid, like spider mites. The necessity of mating to produce any progeny, however, raised questions about the genetic system of phytoseeds, and experiments shows that they have an unusual genetic system called parahaploidy. In parahaploidy, all eggs must be fertilized. Phytoseed eggs that produce females remain diploid. In males, the diploid eggs undergo development, but about halfway through embryogenesis, one half of the chromosomes are heterochromatized and eliminated from the cells, leaving the male haploid for the rest of his life. Some evidence indicates that the chromosomes that are eliminated are inherited from his father. Furthermore, the reproduction of predatory mites requires food, water, and places for the predators to lay egg and live or hide. This requisite can be met by using the appropriate host-prey combination or in an artificial system. The following are some of the methods for reproduction of predatory mite that are done through research. First is rearing on live corn plants. This requires a small production area where multiple 4 inches potted corn plants serve as bunker plants. Bunker plants are a relatively new pest management method that has been used successfully in many commercial vegetable and ornamental operations. Plants are inoculated with pest mites that are specific to the corn plants. The next one is brewing on live ornamental pepper plants. Research conducted by Lance Osborne, PhD, has demonstrated that several ornamental pepper plants serve as excellent hosts of ambitious Siberian predatory mite, even without the introduction of pest insects or mites. Pepper plants inoculated with predatory mite are planted among plants attacked by chili thrips, thus providing a continual supply of beneficial to keep pest mites population low. This system is also effective in controlling whiteflies, broad mites, and other thrip species. Next is rearing on cut foliage. Predatory mites are raised on the pest mites that are specific to the corn host plants. Cut corn plant foliage is periodically placed within cages that contain pure colonies of the host-specific pest mites. These leaf pieces allows for the mites to build large population to be used as food for the predatory mites. Last is rearing on pollens. Nucellus californius and ambitious Siberian predatory mites can be reared on any number of commercially available pollens or field-collected crepe myrtle and cattail pollens. The rearing units are cages made of plastic sandwich boxes or storage containers. The sexual and asexual reproduction in predatory mite. Life cycle, reproduction, and genetic system of predatory mite.